So hello guys, I decided to make this short review before the exam. So I'm going to go over some of the concepts of chapter 3, including a little bit of chapter 2 on molecules. So we were discussing in class that all molecules or that are part of life, right, have like an important atom that it's the center, is the core of all of these big macromolecules of life, right? And that is going to be carbon. And when we're talking about macromolecules, let's remember that we're talking about proteins, we're talking about carbohydrates, we're talking about lipids, and we're talking about nucleic acids, right? So all of these guys are pretty much what we call macromolecules. And the important component of these big macromolecules is that these ones have right at the core, right at the center of those molecules, the atom that is mostly carbon. So that's why everything in biology, biology is the study of life, but biology has to have like this organic component, and that's going to be this important element right at the core of all of these important molecules, right? So let's talk a little bit about carbon, right? Carbon has, right, 6 electrons. We have 2 electrons right there, but these electrons are paired right there on the first shell. And if you guys remember from chapter 2, right, the inner shell of those electrons, right, can only have up to 2 electrons. But in this case, these guys are going to be paired. And right here on the outer shell of these electrons, right, we have 4 unpaired electrons, right? And this is so important because unpaired electrons, if you guys remember from chapter 2, right, represents the valency of that particular atom, right? And valency simply means the total number of electrons that are unpaired, meaning it's only one of these, like in comparison with the inner one that you see, they're paired, so that's good, right? But these guys right here, they're completely unpaired. That means that the valency of carbon is pretty much 4, and that allows the opportunity to create 4 different types of bonds with other different molecules or other different elements, and start making different molecules and arrangements of different molecules based on those type of unpaired electrons, right? And pretty much all of these macromolecules is based on that unpairing of electrons, right? That it allows it to have like such a variety and diversity of molecules and rearrangement of those and making different types of covalent bonds. So we mentioned about covalent bonds, right? Covalent bonds. So covalent bonds, just remember that is just if I go ahead and pair a hydrogen, which only has 1 electron on the outside, and I decide to pair it together with their carbon, right? They're going to be sharing that electron right here. I'm going to put like hydrogen right here. So we have that paired pair of electrons right there, like joining the carbon and the hydrogen right on that structure, right? So that means that we have other 3 possible opportunities to connect that carbon with other elements, right? So this will be covalent bonds. And also let's remember that covalent bonds can be split into non-polar covalent bonds, and also the polar covalent bonds, right? Covalent bonds. The non-polar type of covalent bonds are going to be if I put like hydrogen with another hydrogen right here, right? It simply means that when they're sharing electrons, the electrons are going to be completely symmetrical. They're going to be right in the middle. So they tend to be a little bit more stable, right? So if I go ahead and put another, this will be an example of a non-polar covalent bond. So remember, they are symmetrical. And they tend to be pretty much more stable in comparison with other type of bonds, like the polar ones. If I put here the perfect molecule to compare always to put as an example of the polar molecule is always the molecule of water, right? So let's remember that a molecule of water has an oxygen right here, and we're going to put the 2 hydrogens right here in the bottom. So because of that polarity and that some of those weak electrical forces between these hydrogen atoms right here, this molecule tends to be a little bit more dented, a little bit more bent. But the polarity of this molecule resides that oxygen tends to be like a very electronegative atom. Every time that you put oxygen and connect it with another type of covalent bonds, the electrons will always tend to pull towards the oxygen atom right there. So we're going to connect the electrons right there, very close to that oxygen atom right there. So because of that, if you notice, there's not going to be a symmetry because between the covalent bond between hydrogen and oxygen. It's going to be polarized. The electron is going to pull towards the oxygen instead of being either in the middle or pulling towards the hydrogen right there. So you're going to see that this is going to be asymmetrical. Asymmetrical. This is a polar one. Okay. So what is going to occur is that because we're pretty much putting the electrons more towards the part of the oxygen, we're going to have like a delta negative right here. This area is going to become more electronegative, and we're going to put right here on the hydrogen a more positive delta right there. So the behavior of this molecule is going to be completely opposite, right? We have the negative side and the positive side right there. So it's going to become polarized. So this is very important. We're making other type of bonds with molecules because I think that the behavior of, or the location of the electrons, if you notice, is going to dictate a lot of how that molecule is going to behave. For example, in the cases of nonpolar covalent bonds, we already mentioned that electrons tend to be symmetrical. It tends to be a little bit more stable than the oxygen, the water molecule, right? So that means that when we're talking, for example, about energy, right, in terms of potential energy, these type of bonds, they can have a little bit of higher potential energy in comparison to the polar one. Simply because these bonds are a little bit more harder to break. They're a little bit more stable. A good example of this type of bonds also will be like fat molecules, right? Like lipid molecules. They have a lot of these also nonpolar covalent bonds. So they have like a higher ability to store energy that later on can break away and pretty much release thermal energy or be converted into a different type of energy. But in comparison, for example, water, right? We know water is the perfect solvent. And the beauty of water, of this molecule, is that, for example, if you go ahead and put your table salt, if you guys remember the experiment of the Alka-Seltzer in your in the lab, right? The one that took the longest in the lab was the one that had salt in it, right? Because it became like all saturated. So talking about bonds, right? We're going to go now into ionic bonds. So I can put the example and go back to this example of the molecule of water. So we discussed a little bit of the covalent bonds. So remember, those are sharing the electrons on the outer shell, so they become connected with other atoms. And once you connect 2 atoms or more, boom, you already have like a molecule right there, right? Then we have the other type. We have the ionic type of bonds, right? And this is the perfect example where we're going to use, and we're going to be talking about table salt, right? Table salt. If you go ahead and put like 1 tablespoon of table salt in a cup of water and you start stirring into that salt, you're going to see that it's going to spread. It's going to just dissolve beautifully, right? Until you don't see any traces of that table salt. And going back to that example, right? Sodium, for example, what happens here is that in this case, they are not sharing the electrons like in a way like covalent bonds, but on the opposite. For example, sodium is going to get rid of one electron and it's going to pass it to the chloride, right? So by sodium getting rid of one of its electrons on its shell, right, it's going to automatically become more positive, right? In the case of chloride, when it takes that extra electron from the sodium, let's just say that it accepts it, right, it's going to become negative, right? So every time that you see an ion with a plus sign or negative sign, like sodium, like potassium, like chloride, that means is that you're finding it in its ionized form, right? So in the case of table salt right here, right? If you can see the plus sign, there. We have table salt. This is going to be an ionic bond. So we're going to go ahead and dissolve it like in your lab experiment in water. So we have like a positive side of the hydrogens and we have the negative side of the molecule of the oxygen in the water, right? So what is going to happen, how it's going to dissolve is that every time that you have this molecule of sodium, it's going to go and attach to the negative side of the electron. So it's going to come and form bonds with the oxygen. And the chloride is going to come and join every time the hydrogen, which is the opposite, right? The polarization of it. So that's a way that actually solutes are able to dissolve like in water. And water is like such a perfect solvent for this type of molecules, of solvents, right? So if you notice, going back to the example of the Alka-Seltzer, right? That one was the one who took the longest, right? And you think about it, is because all the bonds that were able to be connected between sodium, table salt, and water were all saturated. Just imagine that like all the parking lots were completely saturated. There was nowhere for the Alka-Seltzer to find like a parking slot, right? And connect with the water. So it took longer for the Alka-Seltzer to dissolve, right? Because it was trying to find like a place to make a connection with that molecule of water and start to have like these reactions and release CO2 and all the reactions of that Alka-Seltzer reaction. Okay, so we have this type of bonds, covalent bonds. We have the ionic bonds. Let's talk a little bit more about the molecule of water. So I'm going to delete this. And let's remember that right here we have a covalent bond, right? But the way that actually a molecule of water connects with another molecule of water, I'm going to delete this portion right here, right? To make it better, is that I'm going to create hydrogen bonds, right? So these are like weak interactions between molecules of water. They're very easy to break, then very susceptible to like temperatures, for example. So we have the negative of the oxygen right there. So it's going to connect with another hydrogen. So we have another molecule of water right there, right? Connecting the positive with an electronegative side of that oxygen through a hydrogen bond. So remember that the way that you connect molecules of water will always be through the connection of those hydrogen bonds, right? So right here, if I have an H, I'm going to be able to connect with the oxygen of another molecule and be able to connect it like that. And I have like another hydrogen bond connecting one molecule with another, right? Over here, I can make like two connections of that oxygen because of those electrical spaces. So I'm going to put another hydrogen with an oxygen right there, and we have like another, hydrogen bond right there. So if we go ahead and start putting these molecules of water in boiling temperatures, right, like high temperatures, you're going to see that the molecules, they're going to start kind of like to break apart, right? They're going to start moving like crazy, like everywhere, right? But we're going to be breaking actually all of those molecules, those hydrogen bonds between them, that allows the molecules to break apart, right? And start moving around each other and increase the movement, the kinetic energy of those molecules in that higher temperature. Also, the molecules can become closer to each other, right? So the density of the molecules are going to be higher. If I go ahead and do the opposite, put all of these molecules, you have the hydrogen bonds right here, right? And put them in a freezing point of water, what is going to happen is that all of these hydrogen bonds, they're kind of like gonna start to spread against each other, like holding hands, but spread and form a beautiful lattice, like crystal lattice between them, but spreading together. So as they spread apart from each other, the density of water is going to become lower, right, in comparison with liquid. So that's why if you go ahead and put ice, a couple of ice cubes in your water, the ice cubes are going to float because the molecules, the density is lower, the molecules are spread apart. So pretty much, let's just say that the weight is a little bit lower, right? The density is lower, so they are able to float in water. And that's why ice floats in water, right? Which I think is pretty cool when you are able to understand how the electrons tend to dictate the behavior of the molecules, participate in the connection of molecules, to the polarization, to the making of other new bonds and new molecules altogether, and play a big role in the behavior of molecules, right? So I think this is regarding quickly water, the bonds that we discussed in class, like a quick review. And that's good. So I'm going to jump from here back to carbon. Let me go ahead and erase this. I'm going to jump into proteins and amino acids, right? If you have any questions, make sure to post them below so I can answer them that you would like me to clarify, or maybe include in other videos that you would like me to discuss. That will be great and very helpful. This is the first time doing this video, so I'm trying it as a test. All right, so let's go back to macromolecules. So we mentioned we have macromolecules, and we're going to make a stop at proteins, right? I'm going to delete this hydrogen and this bond right here, because we need that hydrogen, that carbon right in the middle. All right, guys, so going back to macromolecules. We have macromolecules, so we're going to make a stop here at proteins, right, at the first one. So we discussed that carbon has 2 electrons right there, but it has a valency of 4, right? And we are able to connect the carbon with other molecules or other atoms, right, based on the ability of those unpaired electrons. So we're going to go ahead and I'm just going to focus on those 4 electrons. So let's just imagine that we have the carbon right here, and we have 4 unpaired electrons, okay? And talking about proteins, proteins are simply a polymer made out of monomers of amino acids, right? So if I put one monomer right here of amino acid connected through a peptide bond to another monomer of an amino acid, right? And then I put another one, that's another monomer, that's another monomer right there. So this will be a polymer. So macromolecules in general tend to be polymers made out of simple monomers that you join them together. This is one monomer, another monomer, another monomer, right? All of these together will be a polymer. A polymer. And this is a monomer. So the process of adding a new monomer to this polymer will be the process of polymerization, right? So that will be simply like a very simple way of explaining that a protein is simply a macromolecule. It's a polymer made out of amino acids, right? And that's very simple. Okay, so we briefly mentioned that carbon is at the center. All of these are important molecules in life, right? So we have the valency of carbon, we had 4, so we're going to focus on one simple amino acid right there. Let me put it a little bit over here, because So I want to be able to connect it to and explain something better. I'm going to put it here. Now, in order to put, I'm going to put first the non-ionized form. Remember that ionized, we talk about the table salt, how salt was able to get rid of one of the electrons and pass it on to the chloride, right? So it became like different electrical charges on that table salt. Sodium becoming more positive, chloride becoming more negative. So have that in mind. Right now we're going to talk about the amino acid in its non-ionized form. So we have that non-ionized form. We're going to have carbon. We're going to put right here on the top one molecule, one atom, one hydrogen atom right there, and one proton on the top, right? And then right here we're going to put one amino, one N with 2 hydrogens right here. And this is going to be NH2 first. Here on the other side, I'm going to put a carboxyl group right here with oxygen. And right here, we're going to put one hydroxyl group on this side. So we have the carboxyl, and we have carboxyl, and we have the amine. And right here on the bottom, we're going to put a residual group. So technically, this will be the basic structure of an amino acid on its non-ionized form, okay? We're going to have the amino group, the hydroxyl group, the carboxyl group. And right here on the bottom, we can have up to 20 different type of residues that we can attach, hence giving us the ability to have up to 20 different types of amino acids, right? So we have all that much diversity to put together. So every time that I'm like talking to the students, you're going to see that I always mention the analogy of Legos, right? So I always think of proteins like Legos. I like all those big figures, and each one of these Lego figures is going to be an amino acid, right? So if I give each one of you, have a different type of amino acids, right? So if I give each one of you a different set of Legos, of colors that have like 20 different types of Legos, each one of you is going to come out with a completely different type of figure and arrangement of those molecules. So you imagine how many types of molecules we could form with different types of amino acids when you put them together, right? If we go ahead and put like a couple of them together, let's just say 20 Legos, right? 20 amino acids, we will have like what it would be like oligopeptides, right? Oligopeptides. That would be kind of like less than 50, joining less than 50 amino acids. Some peptides are as small as just 2 single amino acids. If you put 2 single amino acids together, you already have a peptide, and that's very simple, right? So they can go from 2 up to 50 different type of amino acids. So imagine all the different molecules, and of those 50 amino acids, putting them together, a different arrangement can already give us like infinite number of possibilities of making different proteins, right? If we go ahead and put more than 50, right, polypeptides, you can go and just imagine over 50 amino acids, up to thousands, you know? So imagine all these big chains of amino acids arranged together. We can have like just an amazing number of billions of possibilities of different type of proteins that we could have. Okay, so this is exactly the diversity of those 20 that gives us to have like such an important diversity on life, right? So think about the proteins. We have the hemoglobin in our red blood cells, right, that act as transporters of oxygen from the lungs to deliver the oxygen to all of your tissues. Then maybe of the keratin in your hair, the elastin of your lungs that allows protein, that allows your lungs to expand as you breathe in and exhale in air. Think of the different types of proteins that you have in your skin. For example, we have different types of proteins that we have in your muscle cell, like actin and myosin fibers, right, that allow the movement, allow us to move and have that contraction of those bands in your muscle tissue. We also have in your immunoglobulins, right? For example, if you have like a virus or an infection, we have this whole immune system that have these immunoglobulins that can go ahead and attack and defend us from getting sick. They also have released chemokines that act like a communication system that if some cells cannot get rid of a virus, they're going to give a call to others to come and assist and get rid of that virus, right? So peptides can have like a functioning structure in movement, in signaling, in self-defense as part of the immune system. So they can do so many things. So all of our body is very important to have our system and proteins made of these amino acids. Now, we're talking about these amino acids, right? And we're talking that we have like a basic structure of that non-ionized form. Now, that non-ionized, we briefly mentioned also in class that when you put this amino acid in water, for example, right? Or if you put it in our blood, and water has like a pH of 7, our blood is very close to it, right? With a 7.34, 7.35 to 7.45. I'm just going to put that range, right? So our blood is very close to neutral. When you put amino acids in a neutral environment, they're going to start to become ionized, right? They're going to exchange protons. And that allows us to be more stable in that environment and also carry out, to connect with other molecules and carry out all of our functions that are vital in life. So let's go ahead and put this blood in an ionized form. So what is going to happen, very simple, is that we're going to put this hydrogen, like sodium, right? Got rid of a proton, right? We're going to send this hydrogen to the other side. We're going to connect it right there. We're going to polarize it right there. It's going to become positive. And the oxygen right here, it's just going to become, it's going to disappear. And we're going to have like a negative side on it. So the carboxyl part of it, NiH, NH, and NH2 is going to become NH3, right? NH3 now, because now we have 3 hydrogens right there. And we have the carboxyl group with the O that is becoming a little bit negative. So this is going to change like a lot of things. The carboxyl groups, being like that, will tend to become a little bit more acid on that environment. The amino is going to become and behave a little bit more like a base. So we have the acidity of one side and the base. So you're going to see on, if you go back to your book, and you're going to see we have like different type of residues, right? Of 20 different type of endings that can give us 20 different types of amino acids. And these ones can become acid, base, they can become polarized, nonpolar, right? Electrically charged, not charged, or not charged, uncharged. So based on that, it's going to dictate how this whole amino acid is going to behave. In general, is it going to be a type, an acidic type of amino acid? Is it going to become like a basic? Is it going to become like a nonpolar? Is it going to become charged? And it's going to dictate the behavior of that particular amino acid. So in this case, we have it ionized. An ionized type of amino acid now, okay? Because we were able to make that exchange of protons from one side to the other one. Okay, so we're saying that the primary structure, right, of an amino acid or a protein is a polymer of these amino acids. So I want to go ahead and make a connection of another amino acid right there. So I'm going to put the exact same structure right here. It's the same color? Yes, it's the same thing. We have the residue right here, and we have the amino with the 3 hydrogens right there. Okay? So we have one amino acid, and now we want to be able to connect it with a new amino acid right there. So we want to be able to create a peptide bond, right? So one amino acid, it's connected to another amino acid through the formation of peptide bonds. And that's what we want to do right now. So in order for that to happen, every time that we connect one amino acid to another one, we're going to go, something very interesting is going to happen, and that is the process of condensation. And in condensation, what is going to happen, right, because we want to connect this group, the carboxyl side of one amino acid, with the amino group of the new amino acid, and we want to join them together to create that peptide bond, right? So what is going to happen is that we're going to get rid of that oxygen of that amino group, and we're going to get rid of 2 hydrogen atoms from the new, from that amino group. And if you notice, what molecule is that, right? Every time that we're pairing one amino acid to another one, we're going to release one molecule of water right there. And that is called a condensation reaction. Condensation reaction. So every time that we pair these amino acids, you're producing water. We're releasing water, like in that process. So as I'm able to connect those two, release those molecules of water, I will be able now to make a connection between the carbon and the nitrogen of the new amino acid, right? And right here, finally forming a peptide bond. And that peptide bond is going to be simply a carbon-nitrogen covalent type of bond, right? So peptide bonds, remember, is going to be forming between the carboxyl, the carbon of the carboxyl group of the previous amino acid with the nitrogen of the amino group of the new amino acid. And there you go. You have your peptide right there, and you have your first polymerization reaction. And in the process, we were able to release water through that molecule through a condensation reaction. Okay, so very good. Let me go back now, and we're going to talk about the different structures of amino acids until we are able to make a protein. Give me a second to go ahead and delete that. So we have different types of structures of amino acids of a protein. We have the primary, the secondary, the tertiary, and the quaternary. I'm going to delete also this information right here. Let me just quickly clean the board. It's a little bit sticky, not to lie. There we go. Okay, so after that long pause of erasing the board, let's talk about the different structures of a protein, right? In order to actually call it a protein, a polymer of amino acids, that protein needs to be folded and have the perfect shape and everything arranged nice in place to actually become a protein. It is very important for the protein to have the proper structure in order to work and function as a protein. If that protein loses its function, its shape, it's going to lose its function. So the concept of structure and function is very important when it comes to proteins. So let's go ahead and cover the primary structure of an amino. And we're going to be talking about 2 important concepts right here. So we can understand how proteins are being formed and what is happening in our cells and where is that taking place. So taking a big pause, right? We talk about the central dogma of biology, right, in chapter 1. And that's simply when we have our genetic information, and from that whole genetic information, we are able to get information from one gene, right? One recipe. All of your DNA is like a big recipe of life, like a big USB that has all the recipes to make all the proteins that you need in your body, right? All the information. But one single gene is translated to create a protein, right? So that's going to be the central dogma in biology, and both are taking place. So imagine this is going to be happening in our cells, And we have our DNA right here inside the nucleus, right? So we have our DNA right there. And we need to begin with the primary structure of a protein, right? For that to happen, right, then we have the DNA, all the information, but in order to make proteins, we don't need the whole DNA. We just need one single gene, information of one gene, to be translated into an amino acid, right? So we have the gene right there, and the gene right there, it has to be passed into an RNA, transcribed, and this is a transcription process right here. So we're going to call this a transcription process and the RNA. And we're going to talk about more in detail later on in chapters on, but RNA has to become messenger RNA in order to successfully be able to exit the nucleus. This is the nucleus of your cell. Just imagine all of this is one big cell. So it's finally being to exit as a messenger RNA. So right there in our messenger RNA, we have these macromolecules that are called ribosomes, right? And these are going to be our ribosomes. So as the messenger RNA exits mRNA, it's going to enter this ribosome. And the ribosome is going to translate. Imagine I give you one piece of information in a different language or images, like a coding, right? And I give you, this is the code. So you have to be translating, I don't know, the circle means it's an A, right? A triangle means it's a B. So I give you that information, and you're going to be translating the message that I give you into the proper amino acid sequence. So that's pretty much what the ribosomes are going to do. They're translators of genetic information. So as the messenger goes in, we're going to have the primary structure of an amino acid. So right there, we have the primary sequence of information, right? So right here, the primary structure is just going to be based off all of these big polymer of amino acids joined together. And let's remember that primary sequence, just imagine that's the information, simply linear sequence of information, right? Right here, you will always find the amino group on this side, 3, the positive sign, and you will always find the carboxyl side on your right, or the left of the amino acid structure, the polymer. And right here, the important thing that you guys need to remember is on the primary structure, you're going to find, and it relies on those peptide bonds, right? So that's it. It's very simple. Primary structure is information coming out of your ribosome, and is giving you the whole sequence of information, the arrangement of letters. If this is a methionine, if this is an arginine, if this is tryptophan, if this is, I don't know, methionine again, if this is lysine, this is valine, and so on, right? The different arrangements. So now, and this is going to help you when we talk about the chapter of the cell, because we're going to go through the different organelles that assist in this process. So it's helping you ahead of time. So let's go ahead and move on with the secondary structure, right? On the secondary structure, we're talking that proteins need to have like a perfect shape in order to function each role as a protein, right? So the proteins are not going to work like that. So we need to start folding it, right? So we have right here an organelle that is called the rough endoplasmic reticulum. Don't worry about that right now. I'm just guiding you through the process, right? So this information is going to go right here in the rough endoplasmic reticulum. And what is going to happen right here, right? I want to put it because I have a characteristic of it to have all the ribosomes and all the chaperones. We're going to have a lot of molecular chaperones and other proteins that are going to assist in the folding process of this sequence. So we're going to start to rearrange, and just imagine that we're going to fold this one either into alpha helix structures or beta pleated sheets, right? And we are able to start folding that protein right here in the rough endoplasmic reticulum thanks to the formation of now hydrogen bonds. So hydrogen bonds as we're folding that, they're going to start kind of like, just imagine like sticking together or clipping together different parts of the amino acids so they can take like different shapes like a helix or like a beta pleated shape. So this is going to happen right here on the secondary structure, right? And that is going to happen on the rough endoplasmic reticulum. So there we go. When we move on to the tertiary type of structure, what is going to happen is that we're going to have here, this is the secondary and the 3D shapes, right? We're going to be able to form kind of like a 3D shape right there and be able to have that final 3D structure of a protein, right? But yet we cannot, even though it has a structure, it cannot be functionally active until it goes to another organelle called the Golgi apparatus. And the Golgi apparatus, when that protein finally enters, it becomes with the final information and shipping address and where it's supposed to go. It has to have like a specific barcode so it can be finalized. Now, a lot of the proteins, once it has this 3D shape, and before I move on to that, because of the different natural residue groups that we have right here, that can be polarized, not polarized, acidic, basic, you can imagine that the 3D structure is going to have different type of bonds right here. So right here we can have polar, non-polar, covalent. We also have the van der Waals interactions. We have sulfide to sulfide bonds, ionic bonds, more hydrogen bonds. So you can come up with so many type of bonds, so we can finally have that 3D structure at the end. And these bonds are going to be dependent on the type of the residual group that you have on that primary form. Now, some of these proteins, they're going to go ahead and move on to a quaternary structure. Not all of them. Once a protein is by itself and it finishes right here, it's going to be, that's it, ended, right? But some molecules, quaternary structure, they're just going to be, we have heterodimers or homodimers, right? Let's just say that there are a lot of proteins that are going to come and join together as a team and form like a bigger macromolecule complex made of different proteins, and they're going to take kind of like a role as a team, like hemoglobin, right? Hemoglobin is going to be made of 2 alphas and 2 betas, right? Or it can be 2 betas, 2 alphas. If it's the same type of 2 alphas, it's going to be a homodimer, right? Because they're the same. If we have alpha and beta, we can have like an heterodimer type of quaternary structure because these have like different shapes. Okay, so that is all regarding the shape. And once a protein here has finalized, a protein is going to get like a barcode. And some of them, we already mentioned, they're going to stay inside of the cell, and some of them are going to be part of the plasma membrane, and others are going to exit and be part of a different role somewhere else. It's also very important to say that right here we have something that are called molecular chaperones. And those chaperones are very important to assist our bodies, our cells, to make sure that our proteins, once they become like structurally and become like a functional protein, they do not lose the function. They do not lose the shape, right? So they're in charge that, for example, if the cell becomes in a state where there's like too much stress, there's lack of oxygen, ischemia, too much heat, these guys are like little supervisors making sure that all of the proteins maintain the integrity and the structure of the protein. Remember, if a protein loses its structure, it's going to lose its function. So it's not going to work anymore, and the cell can die. And of course, imagine our bodies where proteins are just becoming denatured. It's going to have like an impact on our bodies and lead to even to multiple organ dysfunction, right? Because it's not working, our organs are not are shutting down. Okay, now talking about denaturalization, right? So we mentioned that if we put the molecules in water, right, and they have molecules with another molecule of water. They're connected through hydrogen bonds. And if we put them in like boiling water, they're pretty much going to go ahead, break apart all those hydrogen bonds, and they're going to evaporate, right? They're going to split and they're going to evaporate. So the same thing is going to happen with the hydrogen bonds in the secondary structure, right? So right here on the secondary structure, if those hydrogen bonds are broken, that this structure, the helix structure and the beta plate sheets, they're going to start kind of like to become denatured, right? If they break, it's going to become denatured. So that denatured protein is going to break. The structure is going to lose all of its function. Even if you go ahead and heat up the protein on the tertiary structure, they're going to start breaking apart like all of these bonds, and it's going to go back to its primary structure. Okay, so obviously the protein is going to lose its function. So sometimes some of these proteins, thanks to the help of these molecular chaperones, like the heat shock proteins, right, able to kind of like help them, kind of like assist them, bring them back, and help them to get refolded again, right? If the damage is too big, then sometimes the cell can go into apoptosis and it pretty much is gonna go ahead and die. The heat shock proteins are gonna exit, and it's gonna create like such a big chaos in our immune system. But just remember that these guys are in charge, or supervisors, to make sure that all the proteins are being folded correctly, and all the already functional proteins already in your body are maintain their shape so they can function effectively, right? So that will be the So I think that's it for the review that I can think of. So I hope you like this review. Please let me know if this helps, or if there's something particular that you will like me to cover, so to help you learn everything that you need about this course in biology. Thank you guys so much. Have a great day.